Hello people, welcome back to my YouTube channel. I hope you are in good health. I am back with a new research paper explanation for you today. So, let's get started.
Liver regeneration, comprehensive insights into biological and pathological mechanisms. Introduction. The liver, one of the largest and most vital organs in the human body, plays a crucial role in various metabolic, detoxification, and synthetic functions. Its remarkable regenerative capacity has fascinated scientists and clinicians for decades. Unlike most other organs, the liver can regenerate lost tissue, restoring its original size and function even after substantial damage. This regenerative ability is essential for maintaining the body's metabolic equilibrium, making the liver's regenerative mechanisms a critical area of study for understanding both health and disease. Physiological basis of liver regeneration. The liver's regenerative ability is governed by a complex interplay of cellular and molecular events, ensuring that the organ maintains its functional mass relative to the body's metabolic demands. This concept, often referred to as the hepatostat, ensures that liver size and function are tightly regulated. 1. Homeostatic regulation. The liver's ability to regulate its size is evident in various physiological states. During pregnancy, the liver enlarges to meet the increased metabolic demands. Conversely, in conditions like cachexia, liver size decreases as the body's energy reserves are depleted. This adaptability underscores the liver's unique ability to respond to changing physiological needs. 2. Regenerative mechanisms. The liver employs multiple regenerative strategies to maintain its functional capacity. These include proliferation of existing liver cells, activation of stem-like cells within the liver, and even transdifferentiation of non-hepatic cells in certain contexts. Recent advances in molecular biology have identified key signaling pathways and transcription factors that mediate these regenerative processes. Molecular and cellular events in liver regeneration. Understanding the molecular and cellular basis of liver regeneration involves exploring the intricate signaling networks and cellular dynamics that facilitate tissue repair and regrowth. 1. Signaling pathways in liver regeneration. Several key signaling pathways orchestrate liver regeneration. These include hepatocyte growth factor, HGF-c-Met pathway. HGF is a potent mitogen for hepatocytes, and its receptor, c-Met, is crucial for initiating hepatocyte proliferation. HGF is produced by mesenchymal cells and acts on hepatocytes to trigger DNA synthesis and cell cycle progression. Epidermal growth factor, EGF, and EGF receptor, EGFR. EGF stimulates liver cell proliferation through its interaction with EGFR. This pathway activates various downstream signaling cascades, including the MAPK and PI3K/ACT pathways, which promote cell survival and growth. Transforming growth factor beta, TGFB. Although TGFB typically inhibits cell proliferation, its role in liver regeneration is complex. It modulates the balance between proliferation and apoptosis, ensuring proper tissue remodeling. Cytokines and inflammatory mediators, interleukin-6, IL-6, and tumor necrosis factor alpha, TNFA, are critical for the priming phase of liver regeneration. They activate transcription factors such as STAT3 and NF-κB, which initiate the expression of genes involved in cell cycle progression. 2. Cellular dynamics. Liver regeneration involves the coordinated proliferation of various liver cell types. Hepatocytes. These are the primary functional cells of the liver, responsible for the bulk of its metabolic activity. During regeneration, hepatocytes re-enter the cell cycle, proliferating to replace lost tissue. Cholangiocytes. The epithelial cells lining the bile ducts also contribute to liver regeneration. In certain conditions, they can transdifferentiate into hepatocytes, demonstrating the plasticity of liver cells. Non-parenchymal cells. Liver sinusoidal endothelial cells, LSECs, Kupffer cells, liver macrophages, and hepatic stellate cells all play supportive roles in regeneration. They secrete growth factors and cytokines that modulate hepatocyte proliferation and tissue remodeling. Regeneration after partial hepatectomy. Partial hepatectomy, PH, is a well-established experimental model for studying liver regeneration. This surgical procedure involves the removal of a significant portion of the liver, typically 2/3 in rodents, triggering a robust regenerative response in the remaining tissue. 1. Immediate responses. The removal of liver mass leads to hemodynamic changes, including increased portal vein pressure and shear stress on LSCCs. These changes stimulate the release of signaling molecules such as nitric oxide, NO, and prostaglandins, which promote vasodilation and enhance liver perfusion. 2. Extracellular matrix remodeling. Matrix metalloproteinases, MMPs, and other proteolytic enzymes degrade the extracellular matrix, ECM, releasing bound growth factors and facilitating cellular migration and proliferation. This remodeling is essential for creating a conducive environment for liver regeneration. 3. Priming phase. The initial phase of liver regeneration involves the priming of hepatocytes, making them responsive to growth signals. Cytokines such as IL-6 and TNF-A activate transcription factors that upregulate the expression of genes required for DNA replication and cell cycle progression. 4. Proliferative phase. Following priming, hepatocytes enter the cell cycle and undergo rapid proliferation. Growth factors such as HGF and EGF are critical for this phase, driving the expansion of hepatocyte populations. 5. Termination and remodeling phase. As the liver approaches its original mass, regenerative signals are downregulated, and growth inhibitory factors are upregulated. This ensures that regeneration ceases once homeostasis is restored. The ECM is remodeled, and hepatic architecture is reestablished, completing the regenerative process. Transdifferentiation and stem cell dynamics. The liver exhibits remarkable plasticity, with the ability to harness stem-like properties of various cells to aid regeneration. In scenarios where hepatocyte proliferation is inadequate, other cell types can compensate. 1. Hepatocyte cholangiocyte. Transdifferentiation. Under certain conditions, cholangiocytes can transdifferentiate into hepatocytes and vice versa. This transdifferentiation is crucial for maintaining liver function when the regenerative capacity of primary liver cells is impaired. 2. Activation of progenitor cells. In chronic liver injury or severe damage, hepatic progenitor cells, HPCs, also known as oval cells in rodents, can differentiate into both hepatocytes and cholangiocytes. These progenitor cells reside in the canals of Herring and are activated when the regenerative capacity of mature liver cells is compromised. 3. Mesenchymal stem cells, MSCs. Emerging research suggests that MSCs from bone marrow or other sources can migrate to the liver and differentiate into hepatocyte-like cells, contributing to liver regeneration. This potential has significant therapeutic implications for regenerative medicine. Pathological aspects of liver regeneration. While liver regeneration is generally beneficial, certain pathological conditions can arise from dysregulated regenerative processes. 1. Chronic liver disease. In chronic liver conditions such as cirrhosis, ongoing liver damage and inflammation lead to continuous cycles of regeneration and fibrosis. This persistent regenerative effort can exhaust progenitor cell populations and promote fibrotic scarring, ultimately impairing liver function. 2. Liver cancer. The regenerative process can also predispose to cancer development. Chronic liver injury and inflammation lead to genomic instability in proliferating hepatocytes, increasing the risk of mutations and oncogenesis. Hepatocellular carcinoma, HCC, the most common form of liver cancer, often arises in the context of chronic liver disease and impaired regeneration. Therapeutic implications and future directions. Understanding. Liver regeneration's intricate mechanisms opens new avenues for therapeutic interventions. 1. Regenerative medicine. Advances in stem cell biology and tissue engineering hold promise for developing regenerative therapies. Inducing the differentiation of stem cells into functional hepatocytes or using bioengineered scaffolds to support liver tissue regeneration are areas of active research. 2. Pharmacological interventions. Targeting specific signaling pathways involved in liver regeneration could enhance or modulate the regenerative response. Drugs that mimic growth factors or inhibit inhibitory pathways may improve outcomes in liver transplantation or acute liver failure. 3. Gene therapy. Gene editing technologies such as CRISPR-Cas9 offer potential for correcting genetic defects underlying liver diseases. By targeting specific genes involved in liver function and regeneration, gene therapy could restore normal liver physiology. 4. Liver transplantation. Understanding liver regeneration can improve strategies for liver transplantation, including optimizing donor-recipient matching and enhancing the regenerative capacity of transplanted livers. Conclusion. Liver regeneration is a complex, multifaceted process that underscores the liver's vital role in maintaining body homeostasis. The intricate interplay of signaling pathways, cellular dynamics, and environmental factors orchestrates this remarkable regenerative capacity. While generally beneficial, dysregulation of these processes can lead to pathological conditions such as fibrosis and cancer. Advances in understanding liver regeneration hold promise for developing novel therapeutic strategies, offering hope for patients with liver diseases. As research progresses, the potential for harnessing liver regeneration in clinical practice continues to expand, underscoring the importance of this field in both basic and translational research.